The overall goal of this procedure is to capture brain activity and kinematic data from infants by integrating synchronized high-density active scalp electroencephalography, inertial measurement units, and video recordings. This is accomplished by first placing the EEG cap on the infant's head and ensuring acceptable impedance levels between each electrode and the scalp. The second step is to secure inertial measurement units on the head, arms, and chest of the infant, as well as on the wrists of the experimenter. Next, the experimenter interacts with the infant in an effort to initiate an imitation response. The final step is to digitize the 3D spatial coordinates of the EEG electrodes. Ultimately, the acquisition of EEG and kinematic data from freely behaving infants allows the investigation of the neural basis of cognitive motor development and the emergence of social cognition.
The main advantage of this technique over other approaches that investigate social cognition is that we are able to acquire neural activity in action and in context during the first 2 years of life.
This method can help answer key questions in the cognitive neuroscience field, such as what are the intrinsic relationships between developmental changes in brain architecture and behavior.
The implications of this technique extend towards therapy of social cognition problems and autism spectral disorders, because it can be used to study developmental milestones in learning by imitation, how infants process visual actions, and how they engage in social communication.
Though this method can provide insight into understanding the functioning of the mirror neuron system in the early human brain, it can also be